Hi everybody, this is Miss Bentley. It's going to attempt to do a video for your biology EOC review. They're calling it the Georgia Milestones this year. So let's go ahead and get started. At any time, just kind of remember that this is a very quick overview. We went into a lot more detail in class. This is just trying to be a very quick overview of things that we covered in class to kind of help refresh your memory. All right, so the first unit that we'll talk about is the cells. So this is the state standards, what you should be able to do on the test, what they're looking for. They're looking for you to be able to know those organelles, the difference between a prokaryote and eukaryote, including how that cell membrane helps them maintain homeostasis and overall cell reproduction. They're also looking for how enzymes function as catalysts. We'll talk about what a catalyst is, those macromolecules that you guys loved, explaining the impact of water via osmosis and diffusion, and the special properties of it. Also, you're going to have to be able to distinguish relationships of the single-celled organisms and the multi-celled organisms within complexity of those systems and explain the cycling of energy through photosynthesis and cellular respiration. Okay, so we'll start with the macromolecules. So remember we had macro being large, four macromolecules. We had proteins, carbohydrates, lipids, and nucleic acids. Those are the four main categories. Then each of them had a monomer. Remember, a monomer is a single subunit of a larger macromolecule. So you put monomers together and they build polymers or macromolecules. The monomer of a protein is an amino acid. Monomer of a carbohydrate is a monosaccharide. So remember, glucose is one of our big monosaccharides. Remember, mono means one, saccharide is sugar. Anything that ends with ose is going to be a sugar. Lipids, we have two monomers, fatty acids and glycerol. Remember I said, don't give me any lip or I'll kick your fatty acid? Ha ha ha. Insert corny joke there. Nucleic acids, nucleotides. So a monomer of a nucleic acid is a nucleotide. How do we get it? How do these, we need these molecules in our bodies in order to function because they have great functions within our body. So how are we actually obtaining it? We did this with our macromolecule munchie day, and you were able to bring in different food items, and you had to talk about how that food, what macromolecule it provided you. A lot of foods actually do double duty. Proteins, you think your meat, your dairy, your bean products. Carbohydrates, you can have simple carbs and complex carbs. Simple carbs, that quick sugars and that kind of stuff. Complex carbs are going to be things that have a lot of fiber and stuff like that. Lipids, you got saturated and unsaturated fats, and nucleic acids. Well, remember, nucleic acid, that's your DNA. So anytime you're eating any organism, which is a living thing, it's going to have the DNA in it. Um, the big thing about the macromolecules is knowing the overall function. You got to know the monomer and the function. So with proteins, we have several important ones. We talk a lot about proteins throughout the entire year because proteins are such a part of you. You are a walking, talking protein. Your skin is protein. Your hair is protein. Your eyes are protein. Your nails, all structures that make you up protein. Your blood is protein. In addition to all that, as though that wasn't enough, enzymes, which are a really big unit that what we talked about, which remember enzymes help speed up all those chemical reactions that are having to go on in your body. Carbohydrates are used for short-term energy storage. So whenever you are going to go run a race or something like that, your coach tells you to carbo load, eat that spaghetti or whatever to help, give you that short energy that you can burn very quickly. Lipids, unfortunately, if you don't use those carbohydrates that you eat, that's why people get that fat on them. That's not long-term energy storage. So if you don't use up all the carbohydrates that you end up taking in, it ends up getting stored as a lipid into long-term energy storage. And then remember that long-term energy storage for the lipids, it's also good for insulation, and it's also really good barrier. We'll talk about the phospholipids in a little bit. Nucleic acids are really important because they're our DNA and RNA, and they store genetic information and they transmit it via protein synthesis. And so that's going to be our DNA and RNA. Some examples of from proteins are amylase, which is an enzyme. I'll remind you again later. Anything that ends with ACE is going to be your clue for an enzyme. Carbohydrates, remember we had monosaccharides for our monomer, so a polysaccharide, glycogen, starch, cellulose. Remember, glycogens were for humans, starch and cellulose were for plants. Lipids, you have cholesterol and adipose tissue, which is basically just fatty tissue. Um, and we use it for warmth and extra cushion. And then nucleic acids, as I've already said, DNA and RNA. Um, then we'll move on to enzymes. So an enzyme is a protein that speeds up a chemical reaction. Now, how does it do that? It lowers the amount of energy that's actually required. And because it lowers energy, we call these things catalysts. So catalyst is just a fancy word, or it just means makes things happen faster. It speeds things up. Because all of these chemical reactions in our body, it would require so much energy for these things to occur, like breaking down our food or even breathing. All the energy that's required, we couldn't possibly eat enough food in order to do this, the amounts. So we need those enzymes. If you didn't have enzymes reactions, your cells would start dying, waiting for the reaction to occur. Enzymes are both used to break things down and build things up. They're the ones that are putting things together and pulling things apart and recycling all these parts. And they're very specific. They only work with certain molecules, and we call these molecules substrates. The substrates are just the overall reactants within a chemical equation. In this particular case, this picture here, I'm doing a reaction where I am breaking down a molecule. So I have my single substrate that's put here. It attaches to an active site. Remember, active sites are specific to their substrate. We call this the lock and key model, where my key will only fit my door. Right? I couldn't take my key and go open up Miss Murdoch's door or anybody else's door in the school. It'll only work on my door. The substrate will only work on a certain enzyme, the enzyme that it's designed to work for. Once it attaches to the active site, the chemical reaction occurs. Then it's the job that it was doing was breaking the bonds, and I released two products. Notice that the active site did not change. No, this enzyme is reusable. It will be reused over and over and over again. So as soon as those products are released, another substrate is going to come in and attach to it. Now, enzymes are kind of finicky, just like people. If I put you in a room that was 80 degrees, you would all be yelling at me, telling me to turn on the fan. It's too hot. You can't do any work. You can't learn, all that stuff. Enzymes are the same way. Different enzymes have ideal conditions. We would call this their optimum condition. Optimum just means it's best, what it likes to work at. Like your optimum temperature that you like to work at in the classroom is probably about 72 degrees. Not too hot, not too cold. If we look at this graph here, we have temperature along the bottom, and we have the rate of reaction, enzyme reaction. So this is how fast it's working. So the question is, what is the optimum temperature for this enzyme? So the fastest rate would be around this 40 degrees Celsius mark. And you're probably going to be asked some application question like that with some graph analysis. If you look at that link that I sent you, um, for the biology milestones that the state sent out, you can see a couple practice enzyme questions in there. Now, if we do change the enzyme too much, like if we put it in a condition that's just not going to work out, you see where this rate has gone to zero, what happens is we call that the enzyme has become denatured. So what happens is that this active site, it's going to actually physically change to where a substrate can no longer bind to it. And that can not only because of temperature, but that could be because of the pH that it's placed in. Remember, acid versus base, or it could be the salinity level that it's put in. Okay, so we'll move right on over into cells. So remember, there's two types of cells. We got prokaryote and eukaryote. Pro means no. No membrane-bound organelle, no nucleus. They're much simpler, very much smaller. They will have cell walls. Well, not all of them, but most of them do have cell walls. And they have those ribosomes, and they have the genetic material. A big example of this is bacteria. A lot of times questions won't even say prokaryotic cell. They'll just say, I have a prokaryotic cell. I'm sorry, they'll just say, I have a bacteria cell. And you have to remember in your brain, bacteria and prokaryote are the exact same thing. So when they ask you questions about it, you got to kind of go bacteria, pro means no, no nucleus. I'm dealing with that kind of question. Remember, you do. Eukaryotic, you are a eukaryotic cell. Your body cells and all your other cells have nucleuses, have membrane-bound organelles, much larger, much more complex. pretty much every cell but bacteria. Then the eukaryotic cells are then divided into two main categories of cell. We have the animal cell and we have the plant cell. You should be somewhat familiar with the diagram of them. We drew them, we colored them in, we did all kinds of stuff with this. Um, we remember that they have mostly a lot of the same parts and they serve the same functions. But a couple big differences is that a plant cell has a chloroplast. And remember, a chloroplast loves sunlight showers, be having a blast. We have the vacuole, which is very much larger in the plant cell. Remember, though, some animal cells will have a vacuole, but they just won't be large. They'll be very small. But again, that's not all of them, but the plants do have that very large one for that water storage. And then the other thing unique to a plant cell versus an animal is the cell wall. Remember, bacteria cells do have the cell walls as well. So it's unique that the animal cell does not have the cell wall. You got to know the organelles. I recommend flashcards, or they have those quizdoms where you can play those little games with them. Remember, we have the amazing rap. I won't try to sing it to you, but cells, they're made of organelles. Try to pull a fast one, the cytoplasm gels. Yes, I know I tortured you with my singing voice. But anyway, if you just Google I think it's just cell rap, it's one of the first things that come up on YouTube. So you can listen to that over and over again. But real quick, nucleus, the brain of the cell, stores the DNA. Nucleolus, remember O, it's inside of the cell, and it's going to make those all important ribosomes. Ribosomes are very small organelles. Remember, they are one of the ones that are in all cells because they make those proteins. Those proteins. They do protein synthesis. Remember, synthesis is our fancy word to make. So proteins, remember, are really important to us. They're everything that we are. Then we have the endoplasmic reticulum. We have two types, the rough and the smooth. Rough has the ribosomes on it, where they'll again make more proteins, and smooth helps with lipid synthesis. Remember, smooth like butter, rough ribosomes. Smooth ER makes the lipids. The Golgi body, that's like the post office. It ships and packages and gets those proteins out, ready to leave the cell. Lysosomes are like Lysol. They clean up the cell, get rid of dead parts, bacteria, and all that stuff. The vacuole stores water, waste. Both they're found in both plant and animal. Remember, it's not unique to a plant, as you might have learned in 7th grade. Some animals do have it, but in the plant, it's very, very large. Cell membrane, we'll talk about that even more in a second. The cell membrane, remember, is made out of that phospholipid bilayer. It's going to be the outer barrier that helps control what goes in and out and maintains that homeostasis. Remember, homeostasis being the stable environment, your happy place. Mitochondria, a lot of people mistakenly think only animal cells have mitochondria because the plant cells have chloroplast. Remember, both plant and animal have that mitochondria because they both have to make that ATP. They have to make that energy molecule. So this is the site of cellular respiration. The mighty mitochondria makes me energy. The chloroplast is only in plants, and it helps to trap the light, and it does photosynthesis, converting the sun's energy, the chemical energy into sugar for us, and specifically glucose. Cell walls are the outermost part of plant as well as bacteria cells, but not in an animal cell, which gives support and structure to the cell. And it is made out of cellulose in most cases. The centriole is just found in animal cells, and remember, the centrioles shoot out those spindle fibers. We talked about those, and they help the cell divide. Okay, so how do molecules get in and out? Remember, cells, they have to be small, otherwise they can't transport materials across. So the molecules have to get in through that cell membrane. We call them selectively permeable, which just means they control what goes in and out, and there's some structure to that. Remember across here, these little guys right there, those are called phospholipids. So you have a phospholipid bilayer. Remember, bi meaning two layers, so there's two layers of phospholipids. The tails are hydrophobic, which, remember, they hate water. They hate water, so they turn in tail to tail. There's water outside the cell, and there's water inside the cell, so they've turned away, and that forms that barrier. The heads are hydrophilic, which means that they love water. And so together they form that barrier. Then we have those other components in there as well. We have these big purple guys. We have these, there are different types of proteins, and we'll talk about those in a second. We have carrier proteins and channel proteins and receptor proteins and all kinds of little things. We also, right here, this little yellow one with a little squiggly tail, remember those are cholesterols, and the cholesterols, which are fat, they help allow for that bendiness, so it can be fluid. It causes everything to ripple, kind of like the ocean is just constantly moving back and forth. And then we have the carbohydrate chains up here. Remember, carbohydrate, hi, I'm this kind of cell. It helps identify the cell. Okay, so then we have how those molecules are going to move. So we have smaller molecules can move through diffusion, which is when molecules move from that high concentration to a low concentration. So diffusion is high to low. Eventually all the molecules will spread out to that equilibrium. You can think of diffusion like when your mom's baking cookies and in the oven it's that cookie smell, but it doesn't stay in the oven. It permeates up into your bedroom and you're like, mom's cooking some cookies. So that's diffusion. Um, Now, with cell transport, we can have two types. We can have passive transport and active transport. Passive transport is not going to require any energy. It's going to be that type of diffusion where you're moving from that high concentration to a low concentration. You can have diffusion of just the molecules moving straight through the phospholipid barrier, or you can have something called facilitated diffusion. Remember, facilitate means to help. So it's going to get the help of some different types of proteins because these molecules might be just a little bit too big to squeeze in between those phospholipids. Active transport is going to be a type of transport that requires energy. So remember, our energy molecule is ATP. So these molecules are way too big to fit. Plus, they're going to have to go against a concentration gradient. So what they're moving is instead of moving from high concentration to low concentration, they're moving from a low concentration to a high concentration. So they're moving against the gradient. So passive transport molecules move from high to low. So where, like up here, you see that there's more molecules and they're moving across to an area where there's less molecules. It goes with, we also call that going with the gradient. No energy required, and we have two types, facilitated diffusion as well as plain diffusion. Active transport goes from low to high. So here's where I have low molecules right here. They're using ATP to pump the hydrogens across the plasma membrane. Um, and this does require that energy. We got two types here. We have endocytosis and exocytosis. Endo, thinking bringing in, endo coming in, and then two subcategories, phagocytosis, which is where it's taking in a solid, and pinocytosis, which is taking in a liquid. And then exocytosis, think exiting the building, is when it's releasing those larger molecules. All right, osmosis is specifically the diffusion of water from a high concentration to low concentration. So would it be passive or active transport? It's moving high to low, so it's moving with the concentration gradient. So remember, osmosis will be passive transport. Now, when you have osmosis occurring, you have the water movement, what happens is cells will respond to this. And when you place a cell into we have several conditions of this, a hypotonic solution. Hypo, think hypo the hippo, the cell is going to swell, the cell is going to expand. What that means is that the solute, remember solute, salt concentration, inside the cell is at a higher concentration than outside, so the water is going to move in. Isotonic solution, I'm at equal amounts of solute. So the water's just moving equally in and out. The cell's going to look more or less normal. In hypertonic, the way that I remember that, hyper, like really, really hyper people, they jump up and down, they're usually really skinny. So in this case, the water is going to move out of the cell. It's going to move towards the solute that's outside of the cell. Think about like when you pour salt on a snail. Don't ever do that, but you've probably seen somebody do that. But don't you do that. Be nice to the snails. All the water is leaving the snail's body, and they shrivel and they die. Now, just like we said, there's plant and animal cells. If you haven't noticed by now, the ones at the bottom are the plant cells, and the ones at the top are the animal cells. And when I look at the hypotonic, well, my cell, it just got bigger. Remember, it has that cell wall. That plant cell has a cell wall. And plants are actually really happy in a hypotonic state. They love water, right? They can't get up and get a drink of water whenever they want. So they're happiest at this. They call it turgid. So they're full of water. This is their normal condition. This is when the plant's getting plenty of water. Animal cells, however, we do not have that cell wall. We are not happy. We drown. We blow up, right? We bloat. So our cells will actually rupture, and we call that lyse. The normal condition for humans is that isotonic. Plants, this is when they start to look kind of wilted and droopy. And then both of us, plant and animal, we do not do well in that hypertonic solution. We'll both shrivel up. This is where the plant's pretty much on its last leg. It's going to die any day now. It's time to throw them out, kind of thing. And us humans, we also do not do very well with this particular condition. So hypertonic solution above strength, too much solute outside the cell wall. Water is going to move to the salty side because remember the water, the salt can't move. It's stuck. The barrier's not going to let it through. So the water is actually going to move towards the solute. So if you notice here, I have my less solute here, more solute over here. The solute can't move. The solute can't diffuse in order to equal out the concentration. So what's going to happen is the water is going to diffuse, so osmosis is going to occur, and it's going to move over. So that way, per amount of water, they'll equalize so that the concentration per volume of water would be equal. Hypotonic solution is below strength. So more salt inside the cell, so the water is going to go towards that salt and go into the cell. So I remember hyper, the cell shrinks. Hypo, like hippo, the cell swells. Iso boring. It's going to just say equal, normal, equal. Water's going to move equally in and out. All right, so then we'll do cell energy. Cell energy, remember we talked about photosynthesis and cellular respiration. We talked about it for a very long time. Photosynthesis, I remember, occurs in two stages. We have the light cycle and the light independent cycle. The light cycle is occurring in the thylakoids. Remember my stack of thylakoids. Each one of those look like a pancakes. Pancakes make my thighs get big. A stack of pancakes is really great, so I call a stack of thylakoids a granum. And inside those thylakoids are those chlorophyll. And remember, the chlorophyll are the pigments that attract the light. So the light reactions occur there. And then out in the stroma, which was the ooey gooey, remember I made you pancakes, so the syrup was the stroma, and the Calvin cycle will occur there, in which my glucose is produced. Now, once I have that glucose produced, then the mitochondria, either the plant's own mitochondria that it has, or when I go to eat at the plant or eat an animal that's eaten the plant, I'm able to get that sugar molecule, and my mitochondria, the mighty mitochondria, is able to do that cellular respiration. So it takes in that sugar, we breathe in our oxygen, and we're able to make ATP, which then can be used to power all of our processes. I blink my eyes, I bend my fingers, all that's powered by the ATP. And as a byproduct, I release that CO2 in water, which will then in turn be used for photosynthesis to start over again. So the inputs for photosynthesis, CO2, H2O, carbon dioxide, water, and light. And my outputs for photosynthesis is that organic sugar molecule. And remember, plants release oxygen. And then back to the mitochondria again. There's the overall chemical equations. I would probably expect they would maybe have this chemical equation on there. It is kind of like one of the big things that they emphasize that you should know when you're coming out of this. Remember, the reactants are on the left, the products are on the right. Um, if nothing else, you can recognize water, and you could just kind of process of elimination. Okay, products, an input for photosynthesis. I need to remember to water my plant. And you can remember for respiration, I respire, I breathe in oxygen. So in respiration, oxygen will be on the right, and that should help. Maybe at least with some process of elimination if you can't remember all the letters. But I mean, we wrote this down a lot of times. We copied it, we drew pictures about it, and all that other stuff. So hopefully, this little bit has helped you remember. Okay, enough talking for now. That's it for cells. I will attempt to make a genetics video later. Hope this helped. Again, before and after school, I've posted the reviews on my website and other stuff, but in case you can't come see me, I hope this video helped. That's it.